Good morning, afternoon, evening, whatever you are, early morning, 2 a.m., half asleep, whatever you are back, and we are back. Okay, so what we're going to do in this lesson is kind of preface cardiovascular disease. So I need to introduce you to the lipoproteins. We've talked a little bit about them when we talked about diabetes and so forth. We talked about HDLs and LDLs. It may not have made much sense then, but hopefully after we get done with this session, they will make more sense. But basically, lipoproteins are lipid transporters. Lipids, you can't just put lipids into the blood because obviously they will float, just like oil floats on water, and your blood's mostly water. So there has to be vehicles by which we transport lipids around the blood system and get them from one place to the other, and those are referred to as the lipoproteins. So what we're going to go through is there's different kinds of lipoproteins, and they have different combinations of proteins and cholesterol and things like that. So we're going to go through each one of them, kind of introducing them to you, and then when we get to cardiovascular disease, we'll get more in depth as to what their relationship is to heart disease. And then hopefully you can look back into diabetes and look at those, and it will make more sense after that. So let's go through the lipoproteins and introduce you to them, and then we'll go on to cardiovascular disease after that. So let me get this going. Oops. Yeah, whatever. Okay, so lipoproteins are lipid transporters, and they all have the same basic composition. So all lipoproteins are going to have a core of triglycerides and cholesterol esters. And I'll explain what a cholesterol ester is, because you may not have heard that. And then a shell, this outside shell here, is going to be some cholesterol, some phospholipids, and apolipoproteins, which are basically the identification markers. In biology, cell biology, you should have learned that all cells and things have identification markers because they have to match up to a receptor. And so all of the lipoproteins are going to have apolipoproteins as their identification marker, but they're going to be different kinds. So these are the core parts to all lipoproteins. As we go through, what you'll notice is that the composition changes. Some will have a higher percentage of cholesterol, some will have a higher percentage of triglycerides, some will have a lot higher percentage of proteins and things like that. So let's go through with those and look at them. But first I want to talk to you a little bit about cholesterol esters. A cholesterol ester is a cholesterol that's not active. So it's an inactive cholesterol, meaning that you can't really make anything out of it. It's esterified for storage or for transport. In order to be usable, you're going to have to make it into what we're going to call free cholesterol, and we'll talk a little bit more about that in cardiovascular disease. But you can see here on the left that where you normally have a hydroxyl group, you're going to have a fatty acid, basically, that's attached there, and so that's considered an ester linkage, so that's called a cholesterol ester. And then when you lose that and take the fatty acid off and put the hydroxyl on, then this is referred to as free cholesterol. So later on when we talk about free cholesterol, that basically means it's de-esterified. You take the fatty acid off of there. So anyway, so anytime you're in a lipoprotein or you're storing it, then the cholesterol is going to be in the ester form. Whenever it's active, where you can make hormones out of it, you can use it for putting into plasma membranes and that kind of thing, it's going to be a cholesterol and not an ester form. So it's going to be free cholesterol. So let's start looking at the chylomicron. We mentioned these in digestion, absorption. Basically, we said that they are going to be made in the intestinal. So after kind of recalling, after lipids are eaten, especially the long-chain triglycerides. We're going to put them into a mycel. The mycel goes across the unstirred water barrier, releases those lipids into the cell, and then the cell is going to reassemble them into triglycerides and so forth. And then you're going to put all of the fat-soluble substances into a chylomicron in order to put it into the bloodstream. So the chylomicron is going to contain all the fat-soluble things that we, vitamins, fat-soluble vitamins, the cholesterols, the triglycerides, and those things are going to be in there. What I want you to notice is basically that here's the identification marker. It has basically two, a B48 and an E identification marker. Chylomicrons are the biggest lipoprotein. They're the largest ones that you'll see. They originate, as I just mentioned, in the intestinal cells, and their job is to transport dietary lipids from the intestines to the cells, eventually to the liver. And we'll talk about that a little bit. But so anyway, after you eat, then the lipoproteins, the lipids are going to be put into a chylomicron after they're absorbed, put into the bloodstream, and then travel around, which is going to make your blood look a little bit milky if you have a real high-fat meal, which is why, and we'll maybe mention this also earlier, is when you go to get a blood test for a lipid panel, like you're going to get your cholesterol checked, HDL, LDL, they'll normally have you fast for 12 to 14 hours. And the reason is to clear these out, the chylomicrons out, because it takes about that long for them to clear out and everything. And then you don't deal with them as part of your lipid panel, because they could skew the results if you have a lot of them. So you need to fast for 12 to 14 hours to get rid of those here. But let me just show you a diagram here. So here's your intestinal tract, and you have dietary fat. You put it into chylomicron. Notice the size of the chylomicron, but as it goes through the system, you notice when it gets to the liver, it's smaller, and they call it a chylomicron remnant. The reason for that is that within the cells of the intestinal system, you're going to have the ability to produce what's called a lipoprotein lipase. And lipoprotein lipase is going to be able to initiate the activity of taking triglycerides and basically breaking them apart and allowing them to be absorbed by body cells that need them. So the chylomicron's moving down the blood system, and there's some cells that it passes by that need some triglycerides for whatever reason. Then the lipoprotein lipase will be produced. It will go and start the activity of basically breaking down those triglycerides into glycerol and fatty acids, and then they're taken up by the body cells. So by the time the chylomicron reaches the liver, it has reduced the amount of triglycerides that are in it, so it is going to be smaller and be called a chylomicron remnant. And so when it gets to the liver, then it's going to be taken up by phagocytosis, and actually the cells of the liver will engulf the chylomicron, bring it into the cell, and then break it down into usable units. And we'll talk a little bit more about that in cardiovascular disease and get a little bit more idea of what we're talking about. But just kind of visualize that this remnant is going to reach the liver cells. The apolipoprotein identification markers are going to match up to a receptor on the cell, and then the cell is going to go through endocytosis or phagocytosis, surround it, bring the whole chylomicron into the cell, and then go through the process of the lysosome is going to attach to it, and it's going to basically break it down into usable parts by the liver. Okay, so that's the chylomicron. The VLDLs are very low density lipoproteins. Basically, they are less dense than the higher density ones, but more dense than a chylomicron. But they are also going to be high in triglycerides. I didn't mention that with the chylomicron, but the chylomicron will be high in triglycerides. So it can be, if you don't get rid of them, it can skew the triglyceride numbers. Not so much the cholesterol numbers in those, but the triglyceride numbers. in your lipid panel. The same thing with VLDLs. VLDLs are going to be an indicator of triglyceride numbers. These are actually checked with your lipid panel, so the fasting will not get rid of those, and so they can be an indicator of the triglyceride levels or how much fat you're eating, that kind of thing. And so anyway, it will be high triglycerides, but you notice it has different identification markers as a B100 and an E. So it's different than the chylomicron. So they will fit into different receptors. A little bit smaller. You notice that the chylomicron is about 1,000 nanometers. It's about 90, so quite a bit smaller. The VLDLs are going to be produced by the liver as a way to get rid of excess fat from the liver. Otherwise, you'll get a fatty liver. We talked a little bit about that when we were talking about protein health concerns. And we talked about getting low protein kwashiorkor and you get a fatty liver. And we mentioned the chylomicron or the VLDLs and that, but it may not have made much sense, but hopefully it'll make sense here, that the liver is going to take any excess fat that it has, and it's going to bundle it up into a VLDL and transport it out of the liver going towards the body cells. And so its job is to transport lipids from the liver to the body cells. Let's look at the diagram here, and you can see here's the VLDL down here in the red circle, and so it's going to come from the liver, and it's going to be high in triglycerides, but it's still going to have some cholesterol and protein, you know, the apolipoproteins, the whole thing. But as you, again, what you notice is here's the lipoprotein lipase that's going to become involved so that you can break down those triglycerides and basically put them into body cells, or, you know, if your regular body cells don't need them, then they'll be put into adipose tissue cells, so fat cells, and then you'll get to see them when you put on your bathing suit. Anyway, so that's the way that the liver gets rid of the VLDLs. Gets rid of fat, is to form a lipoprotein, a VLDL, and then transport them into the bloodstream, and then in the bloodstream, triglycerides will be taken out and put into whatever type of cell needs it, or for storage in fat cells. And this is when we were talking about protein health concerns. We were indicating that the lack of protein will not allow you to make a VLDL because there's no protein to make the apolipoprotein and so forth. So you basically accumulate fat in the liver and do that. Okay, so next we're going to go to the IDL, which basically you see that the VLDL, as it goes around, becomes the IDL. So the IDL is not made by anything. It's not made by the liver. It's not made by the intestine. It becomes, as a result of the VLDLs losing their triglycerides, IDLs become, okay, because they're intermediate densities, so they're a little bit higher density. So the intermediate density lipoproteins are going to have less triglycerides, and so obviously if you reduce the amount of triglyceride number, then their percentage of cholesterol is going to go up. But they're really not a big player in cardiovascular disease and actually a lipid panel, and they will actually be eliminated when you go through the 12 to 14 hour fasting period. They will be taken up by the liver. 2/3 of them be taken up by the liver. The rest of them that are going around will actually be transformed into an LDL. So we'll see that basically. But again, it's a remnant of the VLDL. It's smaller. Again, we've lost our triglycerides. We go from 90 to 40. So we've lost our triglycerides. So we've become a little bit more dense. But they're going to be taken up by the liver, 2/3 of them, and then changed to LDL. So when they run a lipid panel, you really won't see IDLs on the sheet as being a component of that. So anyway. Here's the picture, and again I showed you, here's the IDLs, but you can see the arrow going towards the LDLs and the arrow going back up to the liver. And so again, 2/3 of them will be taken up by the liver, and then the rest of them can be changed into LDLs. And LDLs are considered the bad cholesterol. Okay, they're going to be high in triglycer or high in cholesterol, and you'll notice that they are going to lose one of their identification markers, the E, and only going to have the B100 identification marker. So that's an important indicator there. It's smaller, they had 26 nanometers, so it's a little bit smaller, and it's a converted IDL, but it has a positive function. It's not a bad thing. It's called bad cholesterol, but it's really not bad. It's only bad when it gets into the wrong group, right? Just like your kid. Not bad, you just got in the wrong group. Anyway, but cells need cholesterol. Remember, the animal cells need cholesterol in their membranes. We need cholesterol to make steroids, lipid steroid-type hormones. And so we need cholesterol to survive. And we talked about oxidation earlier in the class, and only when LDLs are oxidized, and we'll go into detail on that when we get into cardiovascular disease, only when they're oxidized are they bad. Actually, if they're not oxidized, they're benign. They're not a problem with heart disease. They don't form plaque. They don't do anything. So they're actually good, but when they get with the wrong crowd, oxidation crowd, then they become bad. So we'll talk more in detail later. But that's basically their job. As you can see, the LDLs are taken up, can be taken up by the liver, and we'll talk about the receptor pathway in cardiovascular disease, or they will take cholesterol to the body cells, which is the normal, which they need to. We need a transporter for cholesterol. But the liver is going to monitor how much LDLs you have. So there's a certain limit, homeostasis of LDLs. If there's too many LDLs, then the liver is supposed to take them out of your blood system. But when we go into cardiovascular disease, we'll see that there are situations that will prevent the liver from being able to do that. So we'll talk more in detail about that. So LDLs, the main function is to transport cholesterol to your body's cells. So that's a good thing. But when it gets oxidized, it's referred to as bad cholesterol. But we'll talk more about that later. Okay. HDLs are considered good cholesterol. As you can see, they're very high in protein. They have different identification markers. They're really small on average, and you can actually make them in your intestinal cells as well as the liver. So let me just go through the scenario of what's being made in case you are asked on an exam what makes what. So the intestinal cells make the chylomicron, the liver makes the VLDL, and the liver intestinal cells make HDL. IDLs are not made. They become IDLs when the VLDLs lose their triglycerides, and then LDLs are not made by anything. They become LDLs by being transformed from an IDL to an LDL. And there's some metabolism going on that does that transformation, because you lose one identification marker. So there's some stuff we're not talking about that does that conversion step. But basically, you could say that the more IDLs you have in your blood system, the more LDLs potentially you could have. So let's talk about this, and I'll go back to that theme a little bit more. But here's down at the bottom, you can see your I keep losing my little arrow. Oh, there it is. HDLs are the good guys, and you can see they take cholesterol from the cells, and basically there should be an arrow that goes back to the liver. So if you can, maybe on your PowerPoints, if you would just draw an arrow from the HDL going back to the liver, then it's going to take cholesterol from cells that have excess. It can actually take cholesterol from plaque that's not quite hardened yet and remove it, and we'll talk about that. And any excess cholesterol that might be in the blood system, because the LDL can, if you have cells that kind of burst and release their cholesterol, it can actually vacuum it up, and you can actually have different percentages of cholesterol and LDLs. So HDLs could counteract that by getting to the cholesterol and so forth before the LDLs do. So it is a good guy because it's going to take any excess cholesterol that might be an issue and transport it back to the liver to get it out of the blood system. So it's considered good cholesterol. You can see just showing the classifications of these. LDLs are considered the bad cholesterol. Good is HDLs. These are only bad. I mean, these aren't really bad. IDLs, I don't know why they put them on the bad, but VLDs are bad because they're going to be an indicator of triglyceride numbers, and triglyceride numbers are now becoming more and more important indicator of heart disease, especially, we get to if people with low LDL numbers, if they have high triglycerides, then that could be an indicator of other problems that could be just as bad. And then LDLs, if they're oxidized, can be a problem, as we just said. There are different sizes of lipoproteins. I don't want to go into too much detail, but what we're going to talk about in cardiovascular disease is that the small LDLs are the worst ones. The buoyant ones or the large ones are not as bad because they cannot penetrate the intima, or the first lining of the blood vessel, and get into the blood vessel in order to form plaque. Because plaque is going to be underneath the first layer of the blood vessel. That's why you can't just scrape it off, because it's underneath the first layer, the intima of the blood vessel wall, and so it's going to embed itself in that layer of tissue underneath. And the small LDLs, and we'll talk about concentration gradients. Remember, kind of have to go back to our diffusion days, where we go from an area of high concentration to an area of low concentration. The small LDLs are able to get through the intima, and because they're high concentration, they will get through easily and then become oxidized, or if they're not already oxidized. The big buoyant ones aren't able to get through, so they just kind of stay in the bloodstream and don't form plaque. So we'll say that the small LDLs are going to be the worst ones that you can have. Okay. I want to go back here and just kind of talk a little bit about this, just because I forgot to do it, is as you hopefully you know, and when we talked about respiration in biology, we talked about how if you are totally aerobic, like you're sitting on your rear end watching football and you're eating a lot, and we talked about how, if you're eating Doritos, those can be turned into glucose, go to pyruvate, go to acetyl-CoA, and if you don't need to make ATP, then it will be made into a triglyceride or a fatty acid. We said the same thing about proteins, that proteins are broken down into amino acids, go to pyruvate, go to acetyl-CoA, or if they can be broken down into 2 carbon units, they go to acetyl-CoA. If you don't need ATP, which means you don't go to the Krebs cycle, you go back up and become a fatty acid. Okay, so what this is saying is, and this is all basically going to happen in the liver if you have excess of these, because everything goes to the portal vein and it goes to the liver, if you remember. Then basically all carbohydrates will turn into fats, all excess carbohydrates, all excess protein will turn into fat, and anything you fat will be fat. Okay, so there's going to be conversion of all this excess into fat, which is going to accumulate in the liver. Okay, then the liver says, well, I have too much fat, so I'm going to have to get rid of it, so it makes a VLDL. Okay, and then that VLDL goes out and gets rid of its and then that becomes an IDL, which 1/3 will become LDLs. So it shouldn't be a stretch for you to visualize that if you eat a lot of carbohydrates, you could increase your LDL numbers, because the carbohydrates become fat, become VLDLs, become IDLs, become LDLs. If you eat too much protein, you can increase your LDL numbers. Okay, so it's not all about fat. It's about excess calories, energy that becomes fat that can increase the amount of LDLs. Because the more VLDLs you make, potentially the more LDLs you can make. So eating too much food can increase your LDL numbers. And a lot of people just think, well, it's going to be in the fat. It's not. It's in the amount of energy that's excess that will turn into fat, then eventually become LDLs. Okay, so I want to make sure that you understood that. So this basically is just a diagram of you know, what these lipoproteins contain. You can see the chylomicron. This is the chylomicron is huge, has mostly triglycerides, and then it has the proteins and cholesterol and stuff. The VLDL is going to be mostly triglycerides, not like the chylomicron, but here. And then the LDL is going to be mostly cholesterol, right? High amount of cholesterol. And then the LDLs or HDLs will be high in protein. So that just kind of gives you a visual of what the sizes are. So I think that's the end. I'll go and think, yeah, okay. So hopefully that'll give you an indication of the players. Next lesson we will talk about cardiovascular disease, and we will use these players and fit them into risks for cardiovascular disease. Okay, so we'll see you next.